Welcome to the BioInsights, where science comes alive.
Think about your body for a second. Imagine that it isn't just a collection of flesh and bone, but rather a bustling microscopic arcade. Literally right now, inside every single cell of your body, there are millions of games being played simultaneously. And these games, well, they're governed by tiny, essential, lightning-fast machines. Today, we're exploring these invisible architects of life. But before we get to the machines themselves, let me ask you a question. If you eat a sandwich right now, what's stopping that sandwich from taking, say, years to digest? Seriously, why don't the chemical reactions in your body take forever? Because the truth is, without a special kind of helper, basic life functions, like breaking down the food you just ate or building up a new muscle fiber, would actually be way too slow to sustain life. Okay, let's dive into this. The answer to that question is the enzyme. An enzyme is a specialized globular protein that acts as a biological catalyst. And in chemistry, a catalyst is simply something that speeds up a chemical reaction. But the real magic here, and the thing you absolutely need to remember, is the last part of that definition, without being consumed. An enzyme facilitates the reaction, but it isn't actually part of the reaction itself. It does the heavy lifting, and then it's instantly completely free to go do it all over again. Section 1. Anatomy of an enzyme. To really understand how they work, let's look at what these vital molecules are actually made of. Because they're proteins, enzymes start off as long chains of amino acids. But they don't stay as flat chains. They fold together into a complex secondary structure, and then they fold even more into a tertiary structure. This complex folding pattern gives the enzyme a highly unique 3D globular shape. We're talking about a literal physical molecular architecture with very distinct electrical properties. And all of this complex folding exists to create one absolutely critical feature, a highly specific area called the active site. And this brilliantly illustrates how the active site functions. Think of the classic arcade game, Pac-Man. The main character, Pac-Man himself, represents our enzyme. Pac-Man's mouth is the active site. As you know, it's a very specifically shaped area. Now, what does Pac-Man eat? Those little pebbles, right? Well, in biology, we call the item that perfectly fits into that active site the substrate. The active site is exclusively shaped for a specific substrate, exactly like a lock in a key. Section 2. Lowering the energy barrier. So enzymes fundamentally work by lowering the activation energy barrier. Chemical reactions naturally require a certain amount of energy just to get started. We call that the activation energy. Without an enzyme, that barrier is extremely high, meaning the reaction is going to be incredibly slow. But with an enzyme present, it chemically facilitates the reaction, drastically dropping the activation energy required. The result is a chemical reaction that happens in the blink of an eye. Here is how this builds into the actual step-by-step process. Step 1, the substrate pebble randomly collides with and binds right into that specifically shaped active site. Step 2, we get something called an induced fit. The active site actually changes its shape just a tiny bit to hold the substrate perfectly, kind of like a microscopic enzyme-substrate hug. And finally, step 3, the chemical reaction occurs. The new product is released, and boom, the enzyme immediately resets to grab the next substrate. But, not all enzymes are doing the exact same kind of work. They essentially perform two opposite functions. First, we have catabolic reactions. These enzymes take a really large, complex molecule and break it down into smaller pieces. Your digestive enzymes are catabolic. They take a big bite of food and break it down into soluble nutrients that your blood can actually absorb. On the flip side, we have anabolic reactions. These enzymes take small molecules and build them up into larger, more complex structures, like synthesizing new protein to build muscle tissue right inside your cells. Let's look at a real-life catabolic example in action. Take normal table sugar, sucrose. It's a disaccharide made of a glucose and a fructose bonded together. If you were to just leave a solution of sucrose in a sealed jar, it would break down on its own, but incredibly slowly. However, introduce the enzyme sucrase and magic happens. The sucrose substrate fits perfectly into the sucrase active site. The enzyme puts physical stress on the molecular bond and, utilizing water, a process called hydrolysis, it just snaps the bond apart. It spits out individual glucose and fructose molecules thousands of times per minute. Section 3. The perfect environment. Now, these microscopic machines are incredibly efficient, but man, they are extremely fussy. Different enzymes require highly specific pH levels to survive and function. Extracellular digestive enzymes in your stomach require a highly acidic environment to work. But intracellular enzymes floating around in your arterial blood, they need a very strict neutral pH window between 7.35 and 7.45. This is exactly why systemic medical conditions like acidosis or alkalosis are so dangerous, because if your blood pH shifts too much, those vital life-giving enzymes simply shut down. And it's not just pH either. Temperature plays a massive role, governed by kinetic theory. At low temperatures, there's very little molecular vibration, which is also known as Brownian motion. Because the fluids are cold and sluggish, the substrate very rarely bumps into the enzyme's active site. But as temperatures rise, the molecules vibrate violently. They move around way faster, meaning the substrate crashes into the active site much more frequently. Enzymatic activity speeds up dramatically as it gets warmer, reaching an optimum peak, which is usually right around normal human body temperature.
But what happens if the temperature gets too high or the pH gets too extreme? Well, remember our Pac-Man analogy. In the game, if a ghost touches Pac-Man, his shape distorts and the game ends. In biology, when enzymes hit extreme conditions, they become denatured.
And denaturation is absolutely catastrophic for the enzyme. Extreme heat essentially cooks the protein, causing something called coagulation necrosis. It's very much like how the clear part of an egg turns white and solid when it hits a hot frying pan. The delicate, complex 3D folding of the amino acids completely unravels. When that happens, the active site changes shape permanently. It is ruined. The substrate lock and key system is utterly destroyed, and the enzyme's catalytic function is finished forever.
Section 4. Real-world impacts.
So how do these biological catalysts actually impact our daily lives? Well, many everyday conditions are tied directly to enzymes. For instance, lactose is a large milk sugar. To digest it, you need the enzyme lactase. If a person's body doesn't produce enough lactase, the sugar passes undigested, making them sick. And yeah, that is lactose intolerance. Also, modern medical science relies heavily on enzymes for diagnostics. When cells die or get injured, their membranes break open, and their specific intracellular enzymes leak out into the bloodstream. A doctor can order a blood test looking for these leaked enzymes to pinpoint exactly which organ is damaged. For example, if they find the AST enzyme floating around in your blood, it's a major indicator of liver damage. The sheer scale of what is happening inside you right now is honestly hard to comprehend. Millions. That is how many times faster these enzymes accelerate chemical reactions compared to if your body just waited for them to happen spontaneously. Millions of times faster. And they do it over and over again without ever being used up. Which leaves us with this final thought. Your body relies on this fragile, lightning-fast biochemistry to digest your food, build your tissue, and power your cells. If millions of life-giving reactions depend on these specific lock-and-key machines every single second, what happens to the human body if just one type of enzyme stops working completely? It's a fascinating and, let's be honest, slightly terrifying concept to ponder.